So this is in a sense, I made this presentation and I made it as non-technical as I could possibly do, to give you a glance of what the underlying ideas are. But from listening to the questions and the talks, actually, I think we are further than I thought we are, which is good. But nevertheless, to set the stage, I want to go back to the central dogma. And I want to, everybody knows this, obviously, that we have a process from DNA to RNA to protein, according to the dogma. And this is mostly concerned with the coding region, which came already up, which makes a relatively small amount of the DNA data that are available. So we have this huge non-coding regions that are a little bit more mysterious. But if we stick with the dogma, point of this is that it is not really the DNA, the sequence of the nucleotides, but it is a formed protein. It's the entity that is being interpreted biologically, right? Because it does have some function that it has some kind of biological action. You can't infer this immediately from the DNA. So even here in the coding regions, you have that. And even if you don't believe me, or if you say protein folding, nobody understands this, it's so complicated, to do this in high throughput applications, you would still have a triplet translation, which has an inherent redundancy. Yeah. So even there, translating into amino acids, even if you equate the sequence of amino acids with a protein, which is doubtful. But even if you did this, you would have this redundancy sitting there. So then we have the non-coding parts of the genome, and that is in the overwhelming majority. Now the non-coding DNA is again translated into RNA, and we do have many important cellular processes associated with actually RNA function itself. Aging, I think we'll have telomerases that act, even the very mechanism that generates the proteins involves RNA. And so the non-coding part is even more mysterious, and the question is, how can we actually understand and interpret the data that we see? And this is the purpose of this talk, to zoom into this interpretation. Here I give a little bit of an overview of the active RNA pieces for the non-coding parts. And then the overall idea is that we have a genomic legislative, and we have an, structural executive. And this is written into the process. We always go from DNA to RNA to the corresponding structures. It's like almost the law of the land, how we work in biology. And the important thing is to reflect upon the central dogma on whether that is really almost like an accident to make the protein, or whether it is fully intentional to actually go through the RNA. And the point that I'm trying to make is the fundamental difference between DNA and RNA. Not only is RNA single-stranded, but also RNA is actually folding upon itself, thereby minimizing free energies, or in presence of other entities minimizing something when they form complexes. There are biophysical principles that are being adhered to, most of which we don't know. But the point is that this process of forming a structure is of fundamental difference in comparison to the sequence of the nucleotides. So let me try to explain. Let me try to explain what I mean by that. So if you typically read a sequence and you focus on the sequence of the letters, if you take this haploid and this forms a molecule, the molecule itself is fundamentally not really the sequence of the nucleotides anymore. It is like it's like a scaffold. It is actually like a contact matrix of some kind where you actually remember who is paired with whom. But base pairs can be created by all sorts of in various ways, right? You can have a U and a G pair if it's a wobble base pair, but you also can have by symmetry a G paired with a U. So there is something fundamentally different, if you will, and coming back to Mike's talk, which I try to tie into this, you have another manifestation of the raw data being the genetic data. To form this molecule is really very different. It's like you can view it as a code word. It could be the outcome of the raw information being processed through an information channel. And that's more the picture that I'm trying to evoke, because I think that's important. Because nature somehow found a way to, by the biophysics of the process, to actually process the information that sits on the DNA. So let's look at our language. How do we do this, right? So this is not my attempt to write an English paragraph, right? So indeed, this is apparently easily readable for you. You can infer the meaning of that. You also see that the upper two rows are hardly matching in the sense of alignments. If I actually look at the letters that are matching, there are not too many of them, but the meaning of these sentences are almost equal. So meaning that our language is actually dealing not with sequences of letters. It's actually, there's a processing involved, which involves grammars, which involves spacing, which involves a concept of verb, of noun, and so on and so forth, even commata, as we will see in a second, that helps us actually to infer semantics of these sequences of letters. And so that is a picture that we are trying to evoke, namely that the genomic data needs something like a grammar to be interpreted. And we are kind of in the quest of theoretically understanding what this grammar is. So how do we get this information from these sequences? And as I was trying to point out 3 minutes ago, this information manifests by folding, if we look at the RNAs, and the folding is actually establishing correlations between positions. Now the point here, connecting to Mike's talk, is interesting, because Mike showed us that reading short words in genomic data is actually allowing us to identify interesting features. It depends on the word length, and it depends on the feature you're looking for, but you are actually able to infer properties from short words in the genomic data. Now, if you look at this here, what you see is that since base pairings involve only 2 nucleotides, that's one of the basic biochemical rules, that you only pair to 1 nucleotide in the basic base pairings. Now it gets more sophisticated when you have tertiary interactions, then you can have multiple bindings, but even if you stick to this simple picture, then you can only be paired to 1 nucleotide. However, that nucleotide can be far away. So in other words, the distance between these 2 subwords, in each of which, so to speak, reversed, right? So this last one here is correlated to the first one appearing here. You have a reversal here. So these distances can be very large because they could appear in the opening and closing segment of a sequence where you have one gigantic stem with a bunch of other structural features enclosed by this. So to connect this to Mike's talk is you can look at the k-words here, but there would be other k-words that are correlated to the k-word you have been looking at. And knowing about these pairs of k-words in your genetic data is going to give you an idea about helices, right? So this hopefully gets the idea across that structure really matters, and that it is less so the actual filling of these positions with nucleotides, but more the fact that this G can pair with the C. So by symmetry, you could then replace, or you can flip the preceding G by a C and the ending C by a G, for instance, or replace it with an A and a U. So you would still establish that relation, and you can satisfy these structural constraints. So the two things I want to point out is the distance between these two things can be really large. So it will be difficult to look for them. And the second thing that I want to point out with this slide here is that it is less really the nucleotide itself, but the correlation that is a relevant factor. So when we talk about comparative sequence analysis, we are looking at the sequence, the genomic data, and we tantamount the genomic data with information. And we have a notion of when things are very close to each other, and we saw even point mutants in those diseases, amazing enough that a single nucleotide change can have this pathological effect. And we say, of course, data are more equal when the nucleotides are basically all the same, and they only differ in a few positions. Here you see a point mutant and the effect of that. Then secondly, in comparative sequence analysis and the Waterman-Smith alignment algorithm that Mike brought up, you also have to insert gaps when you align sequences. Now, I want to make clear that gaps somewhat reflect biological mechanisms that appear in evolution, inserting and deletion segments of the genome in the event of evolutionary trajectories. And this is caught by alignment algorithms by inserting gaps. So in a sense, they try to minimize, or they try to determine the most probable evolutionary relation between two sequence segments. And how you do this is you try to identify subsequences that match very well. And if you can't do this for too long of subsequences, then you insert gaps and try to reconstruct these relations. And if you actually find good alignment scores, you call them evolutionary related. So you see the feature of evolutionary trajectories sitting in there. And the second key assumption is in there that things are equal when they coincide in as many letters as I can achieve this similarity. So in a sense, comparative sequence analysis is about a similarity measure of genomic data. That's what we're talking about. So this summarizes what I just said. We have, by means of the gaps, we take into account the course of evolution and the insertion deletion operations that happen there. And the second thing is, again, the sequence of letters is tantamounted to the information. And then, of course, right, there are further approximations that you undertake. Namely, you form some kind of consensus methods by which you insert the most frequent nucleotide if you have a certain amount of genetic data in order to quantify what is the most likely entry in a specific site position, right? So this is then reflected by the size of the letter that you see at these specific positions, and alignment works very well on these, let's say, derived sequences. Here you have another example that you can actually create whole trees using these methods, and you can get an idea about the evolutionary trajectories. So we usually had the sentence here: if all you pay is peanuts, all you get is monkeys, right? And what we're trying to say here is that with the stipulation that information is tantamount to nucleotide sequence, there come automatic limitations. If I actually wanted to eat grandpa, then that's what I would probably get, omitting a comma. And as you can see, point mutations can really make a huge difference, right? So as we already saw in many examples where point mutants actually induce diseases, those are well-aligning sequences, and you would say, well, I mean, it will be all right, there's only a point mutant, but sometimes this can be really bad. So good alignments are not necessarily guaranteeing me similarity in phenotypic expression. It could be pretty bad, despite the fact that they're very similar, right? And unfortunately, right, flipping this is not going to help you either. Bad alignment is not necessarily an indicator of significant dissimilarity, right? And here I pulled, the human language example where you would have a hard time aligning these two sequences, but they do convey the same meaning. All right, so this sets the stage to what I really want to talk about. So we want to look beyond the sequence level. We do not wish to tantamount information with the sequence of nucleotides. And we want to, if we can, build something like an information theory or information processing for biological systems in a rigorous way, okay? So what do we have? So we have two possibilities. We can either look at the coding segments of DNA, where, as my first slide pointed out, we go through RNA and then through protein. So we have actually a sequence from genetic data to proteins, the executive, if you will. And we have, on the other hand, we have the non-coding side, where we go to RNA, and then being single-stranded, we form structures, RNA structures. And they may have biological function directly. So for conceptual purposes, I want to stick with a non-coding DNA. And I want to remark that in order to understand the non-coding RNA, it is actually quite important to have the fact that you have RNA structures. So I'm saying it's not an accident that we have single-stranded RNA forming structures. And here you see that one RNA sequence can have actually multiple structures, right? So it's not that easy that you have a single phenotype. You actually have a collection of phenotypes, which is, in a sense, an interesting thing. But it is well known that, for instance, that the identical genome, different, say, environmental factors can lead to very different outcomes, right? So we know about these things. And in statistical physics, this is well known that a fixed RNA sequence has actually a so-called partition function of phenotypes, of structures. And these structures appear with the probability that is scaled by their biophysical stability. So it could be free energy, it could be other quantities, but your partition function typically is basically weighted by free energy. So a low energy structure is likely to appear, and there is a singular minimum free energy structure, which is the most likely phenotype for the sequence. However, on that energy level, you may even have multiple structures that are very, very close, right? So we have, on the one hand, for a fixed sequence, many structures. We have, on the other hand, for a given structure, many sequences. So you see we're going very slowly because I want to arrive at something that will resemble information theory, but it's not quite that. So if you figure it out, let me know, because I'm still thinking about it to get my head around it, right? But first I want to show you the inevitable. You do have genotypes. You do have phenotypes. I don't think the phenotypes are an accident. They are the only thing that you can actually measure in terms of biological activity, okay? So I have also told you, and that's important, that the phenotypes themselves are fundamentally different to the genotypes. They have nothing to do with the sequences of the nucleotides. They are more scaffolds. They are more telling you who's correlated to whom in order to be made. And this slide is showing you here tRNA. And there are multiple sequences that make this tRNA. In fact, they're called neutral networks. Those are ensembles of sequences that you can compute. And these sequences all fold into a given structure. Okay? So with these two ingredients, one sequence, many structures, one structure, many sequences, we are led to come to look at the pairs of sequences and structures. We need to consider not only the genotype, but the genotype in combination with the phenotype. All right? And this can be formalized in pairing these two sets, sequences and structures, and then assigning to such a pair, this is a sequence, this is a structure, if I form a pair here, and assigning to this pair a minimum free energy. Call it a measurement how suited that particular sequence is to that structure, but dually, you could also say how suited is a particular structure for that fixed sequence. But you see, it's like, I want to remind you to your high school math is like a scalar product, right? It basically gives you two vectors, and you get a real number. Here I have a sequence and a structure. You get a real number. The real number is just an indicator how well the two of them fit. And Mike and I, right, we are sort of speculating that you could, to some extent, interpret this as an information content of some kind that sits in those two. And then there are various things that you can very easily do here. You can express folding, you can express inverse folding, but the point is that you want to fix a sequence, look at many structures, you want to fix a structure, you look at many sequences. So this pairing is actually telling you the whole story. And it connects also to information theory, where you look at the mutual information of two random variables, right? The issue here is, however, that we're really not looking at random variables, as we're not randomly sampling sequences necessarily, but we could talk about the mutual information of these two random variables. We actually have a concrete sequence and a concrete structure. And what we want to bring to the table to be discussed is to take these pairs as the fundamental object. And the next slides are going to tell you a little bit what this leads to. Now, let me go back and explain to you something. If I weren't colorblind, this would be easier. Let me try this. Ha, that's good, I can see that. So about 1996, John McCaskill discovered that if you fix a sequence, then you can create the space of these phenotypes called the partition function, which I briefly spoke about. Now, with this partition function, you can actually understand the whole folding landscape of that sequence. So this is an important quantity. Now, if you look at this pairing, you're immediately led to look at the dual. Remember that? You have this scalar product. It's called an x comma y. And you can fix an x. And you would actually get a mapping from your vector space to the real numbers. But you can also fix the y. And you would get a mapping, leaving the x open. You would get a mapping from your vector space to the real numbers. So it's a dual space for a reason. Now, this is also suggesting that we have exactly the same thing, not fixing a sequence, but a structure. We must have a partition function of sequences for a fixed structure. And I want to make use. So this is, biological symmetry, aesthetics, mathematics, whatever you choose to believe in. But this is suggested by the very way I formulated this as a pairing, that there must be a dual, and there is. And we can compute this dual actually using recursions, right? In which you essentially take smaller and smaller substructures with subsequences, and you build them up piece by piece, similar to the recursions that you employ when you construct secondary structures. You go with intervals and you make them bigger and bigger and bigger. This being possible because the very secondary structures have no crossing arcs. So what this is doing here, it's creating actually the space of sequences for a fixed structure, each of those sequences having a high information content with respect to that fixed structure. And with this player, and we can actually create this here. These recursions are actually sensitive enough to work within filtrations of Hamming distances. So for instance, I can say I have a reference sequence. Picture this. So I have a reference sequence, a natural sequence. Picture this. I have a natural structure, say a microRNA. And then I can fix myself with these recursions. I can fix myself a Hamming distance, say 10, 7, whatever. And then I can basically sample in that Hamming distance sequences that are very highly likely to recreate the same structure, refold into that same structure. And this sampler works in linear time. Unless your Hamming distance tends to infinity, then this is a different issue. Then it would be quadratic for the constant Hamming distance. But what you see is you can delocalize. Of course, you can look at the point mutants, and then you may as well fold the entire point mutant hull, which is relatively small. But come a Hamming distance of 10 or 20, it is computationally impossible to exhaustively search such neighborhoods. So with this, so these are pictorial depictions of these lovely recursions, but you already saw the multi-indices, so I don't have to tell you more about that. So with this in mind, I want to get back to my description of alignment being really a similarity measure between sequences. So here I'm suggesting that there is one manifestation of this idea associating this fiber of structures to sequences. There is a relation that can be used to quantify similarity of sequences that is fundamentally different in that it does not look in the differing nucleotides. It does actually look at the difference, in a sense, of the associated structure spaces between those sequences. So we are taking into account that the sequences have a certain Hamming distance, right? So if I have this sequence A, this is sequence B, say they're both of length 1000, and I know the number of point mutants, which is easy to determine. Of course, there's a mutational cost to make one into the other. All right? So this is totally clear. But what I'm saying is, aside from this aside from knowing how different the sequences are as the sequence of nucleotides are concerned, I'm saying there is more information sitting there which is accessible very fast to us by creating the structure spaces to these sequence segments. Now what do? I look at these structure spaces, and what I do is I quantify a particular structure. Yeah, this is where my color blindness comes in, right? I have a hard time covering the structures in here, but I guess there are two in here, and they're shared. And now I'm actually looking at these two shared structures, and I remember what their probability of appearing is for the respective sequences. I use all these data to quantify a closeness of those two sequences. And I want to give you a Mickey Mouse example why this matters. Okay, so allow me to flip you through the slides a little bit asynchronously. So picture this. Picture the sequences in question here, these guys here, these two sequences. Say their partition function would be a single structure. Literally anything else is very, very unlikely to ever be realized. So they're really stable. It could be a very long stem or something like that, okay? So if that is the case, for those two sequences to be evolutionarily related is very different because of the presence of that one phenotype that they realize. So why is that? Because in the course of evolution, when that happens, you essentially, this being sequence space, yes? And we just look at the blue dots here. I guess it's blue, I hope. And this is my phenotype here. This is my fixed structure. So if then actually what you're having is those two sequences are part of a network in sequence space. And to be related, I have to kind of cruise along this network neutrally to come from one to the other. Now you may say, why is it important? Well, this is important because actually you will see that when you look at the sites of the two sequences to be compared, that their mutation rates are actually site-specific due to the fact that, for instance, certain sites are very important to maintain the underlying structure. In other words, the presence of the phenotype is guiding the evolution between these two sequences. Okay? So that makes the point that a similarity measure that takes into account the phenotypes is actually reflecting another side. It enhances the evolutionary trajectory that is caught by alignment methods by virtue of gaps. It kind of captures an additional feature. It captures the likeliness of an evolutionary trajectory as it is kind of influenced by the presence of a, say, dominating phenotype that are made up for this example. So I want you to regard this to be on the same level as inserting gaps in alignments, right? So the presence of the phenotypes is leading to site-specific mutation rates, which we know exist. They are a biological reality, and it captures new aspects. Now, with that sampler that I showed you the recursions of, where I can sample sequences in a certain distance, I can actually, for my two sequences that I started with my similarity measure, I can actually very efficiently reconstruct paths connecting two sequences neutrally, meaning paths of only blue dots, where two of them have a very low Hamming distance, and both of them are still blue in the sense of they're making exactly the same molecule. So the reconstruction of such paths is very easy using this particular sampler that I showed. And here is an example that this actually works, where the top And the bottom row are sequences, some kind of Hamming distance. Let this be 15 here. And then the algorithm is actually constructing a sequence of sequences. Each two successive ones of them are either pair or point mutants, and they all realize the same structure. So you can actually understand the trajectory much more precisely. And now I hope I can do that quickly. So keep in mind, here's a similarity measure of sequences, like alignment is, using the space of structures, right? Augmenting the information that I may have by looking at the dissimilarities, the Hamming distance between these two sequence pieces. And now I want you to dualize this. What would you do? You would replace these by sequences. You would replace those by structures. So with this mental picture in mind, you'll understand immediately what this means. So this means that I can immediately, by dualization, take two structures and quantify their evolutionary relatedness by creating the spaces of sequences for both of them, and then quantifying exactly in the same manner these particular partition functions of sequences that I explained how to construct for a fixed structure with a recursion that I presented. So this gives me a new measure of structures. Typically, you would look at structures, you would say, hell, look at the base pairs, count the number of base pairs, or edits, you map them into trees, you would quantify the distance between structures. That's pretty much what we have. What I'm talking about here is actually a distance measure of structures that reflects an evolutionary reality. It's totally different. As I was talking about the distance measure of sequences, that is capturing the fact that they make certain phenotypes and therefore have an evolutionary relatedness. What I'm saying here is there is a measure between structures that is actually reflecting a lot more evolutionary reality than just moving base pairs around and computing an edit distance. So how could you use something like this? Give you an example. Think about a virus, say, that you understand the structure of, say, Ebola, something pretty bad. And now something like this could be used to get an idea of what would be the structural mutants of that structure. What could I expect this thing mutates into, right? For instance, right, I would be interested whether this can become airborne or not. And some clever, structural biologist is going to tell me that I have to look for certain features for this to become airborne, right? I don't know this. I'm from Mars. So he would teach me and he would say, you have to look for this. And then I could basically feed these two structures. Here on the right, I would have a library of structures with these features. And then I would connect these two, and I would see whether there is a chance that I get an evolutionary path from one structure to the other that has a likeliness of appearance. So I can quantify things I could not quantify before. And that comes from this change of perspective of looking sequences and structures simultaneously and treating them as, just two sides of the same coin. All right, so I come to conclusion now. I try to set the stage a little bit for tomorrow's discussion. Wanted to make a point that has already come up twice here, that DNA information is not tantamount. It's not tantamount to the sequence of nucleotides, so we have to infer the biological meaning of these things, right? Then what I'm stipulating is that the molecular phenotypes, the formation of these scaffolds, is really the key in interpreting this. And to go even further, and also Shrikant has brought this up, it's actually a network of phenotypes. It's actually more complicated, right? Because there are multiple phenotypes that somewhat interact, and when you introduce point mutants on the genetic material, you have to understand this, right? And So my stipulation is to look at the sequences and the structures as pairs. And that brings us closer to the information theoretic ideas and the framework that could be applied here. So there's something like, I would view this as a biological interpretation of what we consider a language theory, right? It's probably a hybrid between information theory and language theory, what we're looking at. This is the name of the animal here. And of course, this would shift focus towards methods to have like really fast, high throughput structural information, and have that structural information go into the very process of sequencing, right? The very minimum would be to go through sequence databases and have software tools that can use structural information to infer, meaning that could possibly be gained. In particular, I would expect in non-coding regions this to be a very powerful tool. So the minimum would be new software tools, and if we're really lucky, thinking about the nanopores, we may be able to infer shape information from technologies like this. So this is quite interesting. All right, thank you very much. Any questions?
The example with the mitochondrial DNA, I didn't quite follow. You said there was a signature related to the gap. You had an example on a slide with the mitochondrial DNA, and you were making a point as far as some signature with a gap, the sequence gaps.
I'm not sure, but I can find the. This one? No.
No, the other tRNA toward the end of the talk, maybe. Not that one.
Not that one? What am I missing?
This one, yeah.
This one. Oh, this one. What I'm trying to communicate here is that if you look at sequences and you want these sequences to maintain that shape, and you look at the evolution of these sequences, well, the sequences, of course, have sites, and the sites mutate. Number one, when such a site, a structurally critical site, is actually being altered, then it must be altered in correlation, right? And plus, this leads to different mutation rates at sites. So you have an inhomogeneous spectrum of site probabilities of actually being altered. And the presence of this inhomogeneity, which we know, is actually is an indicator of phenotypic information of some kind to be present, right? Because there would be no reason for those sites to exhibit this otherwise. All right, Mike.
There's a popular book on grammar, and I think I remember the example right. The panda eats, shoots, and leaves. We all hear that in the panda, but if you put the comma after eats, the panda eats, shoots, and leaves. And that complete meaning of the sentence by that one comma. This whole book is full of stuff like that. Just love it.
Yeah.
Could you clarify a little bit what you mean by information content of viewing a sequence and a structure as a pair? You say you view them as pairs, so this has an information theoretic interpretation, but so far as I saw, you didn't actually describe what the information theoretic relationship between the elements of that pair are.
No, we're talking about talking about connecting it to the classical information theory, where you talk about the mutual information of two random variables, right?
So specifically talking about the mutual.
I'm talking about specifically about the mutual information, and I'm trying to make the point that you cannot look at either one of them in isolation, right? So it is actually the two of them that are the core unit to be studied here. Otherwise, it's impossible to understand this. And for a single sequence, you have a bunch of structures. For a single structure, you have a bunch of sequences, right? You have these fibers that lead to the pairing, right? So if you actually were to take the two random variables, one of them being, you know, having the states of all sequences, the other one having the states of all structures, you would be back to the classical information theoretic framework. But unfortunately, or fortunately, right, so that leads to research and us trying to understand and to formulate a theory that adequately describes this. We're not interested in the global mutual information of sequence space and structure space. We're actually interested in what amounts to the information content of such a pair. I mean, the question is whether a natural RNA sequence and a natural RNA structures, what makes them natural? What makes these pairs that you know are realized or evolve in evolution, you know, over different, you know, organism classes, right? What makes them natural? How do they change, right? And that is the key question, right? This is one way of saying what is the information content between the sequence and the structure. And to humor you, we have looked at microRNAs in particular, and we have looked at microRNAs for different organisms. So I can tell you a little bit what happened there. So for low organized microRNAs, and it's a natural sequence, it's a natural structure. So for low level organisms, I think we have worms, say, you actually realize that the stability of that phenotype is at a certain rate, right? Much higher than you would observe in random pairings. It would be at a certain rate, say it's 40%, and then of course it is filtered by distance classes. And by 40%, I mean if you sample this dual partition function as described, a sequence, and you ask that sequence a question, do you fold again into that particular microRNA? And the answer is yes, with a probability of 40%, which is already amazing if you're in Hamming distance 12 or 13 or 14, right? Because if you would take a random sequence, it would never make that microRNA. And then you measure something else. And you measure, in that distance class, you measure the structural variation. You take the sample of the sequences that you make, and then you quantify the structural variation. And I don't want to go into the details of this, but you can do this using statistical physics. So now you go from worm to cow, which is an advancement. And you realize that when you ask the same questions, that your structural robustness goes up from 40% to 60 or 70%, which is interesting. And at the same time, the variability of your phenotype goes up. So what I'm trying to say is, we started asking what is the information content of such a pair, or right? Why do we have to look at the pair? When you look at the pair and you look at the evolution of these pairs, you see that lower organisms are less robust and they exhibit less variability, and higher developed organisms become more robust with respect to the given phenotype and exhibit a higher variability. Now, I'm a mathematician. I don't know what that means. But it makes somewhat sense to biologists who say that having more robustness, of course, if you figured something out, it's good to keep it, right? I buy that. But on the other hand, you also have a higher variability, which means that you can explore phenotypic variance quite efficiently in these distances. So this makes you more flexible in case you are being put under stress. So these kind of questions you can start exploring looking at the framework that I was trying to outline to you. And that's pretty much where this program is. We are trying to find biological footprints, traces that we can use to find the right atom, how to build, for building a theory, right? We want to get it right. And these findings are kind of interesting to note, right?
Let me ask a simpler question. Suppose I have two sequences, and with different probabilities, I suppose, but they map onto the same shape.
Uh-huh.
Okay?
Yeah.
And so then each one of them can be evaluated, those two pairs can be evaluated with respect to their, I don't know, mutual information, some measure.
Yeah.
Let's just say it's the mutual information.
Yeah.
Then is the difference in the mutual information between those two pairs a measure of the difference of the information in the sequences? Is that what you're, is that a fair sort of, I mean, this other stuff is very complicated. I'm coming down to like an incredibly small version.
No.
Of this,
Look,
right?
I'm trying to even
Would this be a difference, a similarity measure between these two sequences?
Allow me to be even simpler. Allow me to stipulate that the structure in those two pairs is equal.
Okay.
So I'm talking about two pairs. They only differ in the first coordinate. So I have a sequence sigma 1, a sequence sigma 2, and they both make the same structure, right? So now this is exactly the pillar for this sequence similarity measure, because now I know these two sequences make that, or with high probability make that structure. Now I can infer to evolutionary trajectories how to come from one sequence to the other. So this means that this way of looking at things is giving me an evolutionary connection between the sequences that I cannot obtain by other means. So it really quantifies the similarity of those sequences once I know they make the same structure. And now, this has nothing to, my simplification is really just that. Conceptually, it's the same thing even if they make different structures, right? So this gives me an idea, and vice versa, right? If I have two structures that stem from the same sequence, that essentially is giving me a relation between these two structures.
Have you considered that there may be another layer that's important to take into account here, in being that perhaps multiple structures could serve the same molecular function and have no sequence similarity whatsoever and no structural similarity, but might actually act to constrain evolution as well?
I completely agree. I completely agree. The reason why I said what I said was, number one, I stayed away from proteins at all, because proteins are very complicated. This is why we have you here. The second thing is, I evoked the partition function of RNA secondary structures. Now, this is just a Mickey Mouse example of saying that phenotypic information is important to interpret the genomic data. It is, as much as the first part is trivial and to most of you completely clear what this is, what I'm really saying is that phenotype and genotype together, and you have this extreme example, right? You take a function, because structure is not equated to function necessarily, right? You can take a function that has many different manifestations on a structural level. They all serve the same purpose, yes? And then each of them is going to give you a whole fiber of sequences folding into it again. And since I have many different structures, I may as well have a random sample of genotypes, all of which generates that function. I'm fully aware that this is possible, and I embrace its possibility because it makes a point. The point is that I believe that the translation from DNA into RNA is not an accident. That's my point. Now, it's sophisticated to use it for translation and then to proteins, but it's almost an accidental thing of higher development, so to speak, to use the proteins as engines. If you run the RNA structures themselves as engines, that makes this point from a logical point of view much simpler, right? Because we don't even have to go through the complicated mechanisms. We have just a folding, and then in absence of knowing anything, we can take minimum free energy folding or partition functions. But conceptually, the key point is that you arrive in a natural way to a phenotype, which I then blatantly equated to function, but I don't mean that literally, right? So you really talk about a function, you really talk about an activity, you really talk about an ability to bind on something that oftentimes is correlated to a certain shape, right? But for me, this is a very, very abstract, very abstract concept. All right, I think we're ready for lunch.